So the last talk in this megafauna session is going to be on something a little different than all the mammals. It's going to be on crocodiles. And once again, our project leader for that project, Andy Halford, isn't available to be here with us today. He's gone on to clearer waters in New Caledonia. But we're going to forgive him for that. We are lucky, however, to have two of our rangers from Lalang-garram Camden Sound Marine Park who actually participated in the research to give us a presentation. So Danny Barrow and Adrian Lane for us.
Oh, you introduced us. That's half our talk is finished. Yeah, so as you know, we both aren't Andy Halford. Andy is over in New Caledonia, having a great time over there, living the dream. I did catch up with him very briefly to have a chat about his data that he collected and just how to present today. And I didn't remember much. All I remember is him saying, "Don't stuff it up." But he used a lot stronger words than that. So Andy, if you're listening, mate, we'll do our best. So my name's Danny Barrow, Senior Ranger for Lalang-garram Marine Parks.
And I'm Adrian. My name's Adrian Lane. I'm the Marine Ranger for Department of Biodiversity and Conservation and Attractions. And yeah, I'm a member of the David McGarry Traditional Owner Group.
We'd just also like to acknowledge the traditional owners, past and present, whose lands we stand on today before we move forward. So we're sort of lucky, me and Adi and myself, we're stuck between the researchers and the decision makers, and you'll sort of see that as we present today. But with the marine park as it was being developed, a few of the planners that are here today put a lot of work into developing a plan that guides us in managing the park as effectively and as efficiently as we can with the resources we have. In that plan, they list down species of cultural significance, species of special conservation value. And with the WAMSI project coming online as it did at the same time, a lot of those key conservation species were already covered. So coral, mangroves, whales, dolphins, all lots of different things, yeah. Turtles, especially turtles. And there was a bit of a gap there with the crocodiles. So obviously with the joint management as well, crocodiles are also very important cultural species. So when Andy started to look at the gaps there in our knowledge, there was about 30 years where our survey hadn't taken place in the West Kimberley. When the last survey they did, which was in 1987, there was 2,500 individuals counted, and I believe that was right across the whole northern Australia. So the species was very much endangered and moving towards extinction. The hunting ceased in 1969, and then move forward, I think. And then, so the hunting stopped in 1969. There was surveys done in 1978 and then again in 69. And what we did was we used NT's data to sort of help set out our survey on what was required, how we're going to do it, what data would need to be collected. So you can see here is just a quick graph that Andy's done up. You see the population growth is sort of quite consistent there. And in 2000, it was declared that the species had recovered from hunting. So in the East Kimberley, there is some survey work getting done. And you can see here the gradual growth sort of population growth from 1980s through to 2000, and then in 2000 something happens and the growth is exponential. So Messels is a bit of a legend, him and his team. They did large-scale surveys in 1978 and 1969 right through Queensland, NT and the Kimberley. They selected most of the major river systems that also had nesting hotspots. Their surveys were very detailed, and we also used the exact techniques and processes they used so we can model our data against theirs.
Yeah, I just want to talk about just with this where Prince Regent sits that sits on top of Camden Sound Marine Park, and then you got the Row River on the very top, which the other survey was done. But I just want to talk about when you come from Broome up to the Prince Regent, and like with the logistics side of it, and where all the marine parks are sitting on top of in the Buccaneer Archipelago area. I mean, the planning and the just to get to some of these spots to do all these surveys, and that it's pretty full on, you know. Just to get even just to have all the resources and the support from the very top down to the bottom to do all this stuff, it just goes a long way to managing the country and the animals as well.
So we were lucky enough, Messels did some great work, as I said earlier, and after selecting the Prince Regent as one of the major river systems in the West Kimberley and the area that we're also doing most of our work, Messels actually drew these amazing maps up, which we found quite handy. You can see they're right down to the kilometres, and you can see right to where he's got to the end of creek systems. So we're able to replicate exactly the sort of survey that he did. This is a sort of the typical area we're working. That's the Prince Regent River for those that haven't been lucky enough to get up there. That's the boat that we operated from, the mothership, and that's famous King's Cascades, where Ginger Meadows was taken by a crocodile many, many years ago. And on low tide, most of the area is, the habitat is these mud flats with sort of sandstone rock up against a couple of local residents. This is the data sheets that we use, the same as Messels, so it's broken down into size category and then whereabouts in the water system that they recorded. So, you know, whether it was in shallow water on the edge, and that was also to keep that data consistent with what Messels collected.
Yeah, and just some of the methods we use to do the counting in the system. So we use a spotlighting system. So what we did with the tide into account to do the actual survey, we did a little mock trial in the first thing in the morning, just to put on a track into the creek systems, and that was just to map out, because we're in uncharted waters and not knowing what's underneath the water and the surrounding areas. We actually had to just put the tracks in and then mark all these things, like these rocks and rock bars and whatever, and then came to the night, and then we had to replicate what we did during the day at night, and do the counting of the eyes. So what we did, we just set, would get in when the tide would come at night. So we'd sit in the middle, and I'd have Denny up the front, and what he'd do, he'd go and if you see an eye sign along the banks or wherever, what I'd do, at a certain speed, he'd guide me into where he wants the croc, or he wants the boat to be in relation to where the croc is sitting. And so what I'd do, I'd drive the boat directed in that direction, and then he'd come up to it, do the measurements, and log it in to where we were seeing it. And then I'd go back to the center. So it's just like a crisscrossing, but it's just trying to get a full coverage of the whole system, so we can get a sort of exact amount of what the amount of crocs in the system anyway.
So spotlighting was the main system that we used to do the count, but there was also an extra project which was the collection of DNA. Andy had partners or friends up in the NT that were also doing a national study, I think, on genetics for saltwater crocodiles. And Andy said, do you think you could get a couple of samples? And we went, we should be able to get one or two a night, no worries, because the standard way of collecting DNA is to actually secure one of the animals using different techniques and taking a sample. And Andy said, no, we actually need 20 or 30 from each river system. So I said, okay. He set a challenge there. So we had no idea we're going to do that, but we looked at different methods, and cetaceans use a biopsy pole for some of their DNA collection. So we thought we'd do the same. However, crocodile skin is very different. The best way to describe it is a dolphin has a skin like an orange. You can peel the skin, where a crocodile has skin like a watermelon. You can't peel that skin. So the biopsy needles that you use on your normal cetaceans, you can't use on reptiles such as crocodiles. So a couple of nights in the shed at home with a few Dremel tools and a couple of beverages to lube up the process, we come up with this biopsy needle, and we tested on the watermelon many a times on a Sunday night, and it worked. and then we took it out onto the croc farm, and it worked. And we got 98% success rate with that, and managed to get the samples that Andy needed. So another tool. So I also just have a little video. Oh yeah, just play a little video just to wake everyone up a little bit.
Oh, did it again?
So we've got the GoPro actually mounted onto the biopsy pole. You can see there, we can see the croc there in the light. We've just picked up the eye shine, and we're coming in close to get a biopsy sample. We take the sample from the muscular part of the tail. It's just a bit of a demonstration of what happens. The crocs don't really know that we're coming up. The front of the boat's all painted, and the light is shone into their eyes, so they can't really see us. Let's stop.
Now you're playing. What?
Yeah, the video is just not happy.
That's all right. Anyway, basically, the animal sort of takes a bit of a snap at the pole. I'm not sure what's touched it, but other than that, it doesn't really even know that it's had a little poke, and the animal recovers really, really quickly. It's only very, very small sort of sample taken. They can survive with arms and legs and things ripped off, so a little tiny poke from the needle is nothing for them. Another system we developed is, or Andy developed with the help of Shane French and the Esri guys, was a method of collecting this data out in the field there around water at night and some of that uncharted water. It's a bit of a tough place to work, and so they come up with this Esri data collector where Andy could collect exactly the data that Messels was collecting, and then once we got back to the boat, uploaded on Wi-Fi, that would be straight into a system here in Perth, and people could see in real time that we were conducting the survey, and that we weren't out just having a good time. But it also enabled a lot of people to analyse the data as it was being collected, and made life a lot easier for Andy when he got back from out of the field. So there's the number of crocodiles we counted. There was 700 that we counted there, a bit over 700. It took roughly 8 or 9 nights, I think, and days. We covered a total of 800 kilometres up and down. So like 80 said, you survey during the day to map your area, and then you go back at night. So pretty intense amount of effort put in a very short period of time. It's got some really good results. You can't quite see there, but there's smaller dots are the smaller crocodiles, larger dots are the larger crocodiles. Andy's analysed a lot of data, but he sort of put this one up as a bit of interest. You can see there the red is the 1978 data, the green is 1986, which is not much of, and then 2015 is the blue. You can see most of the hatchling recruitment is sort of up into the mainstream up in there. If you want any more info on that or a bit more knowledge, you can ring Andy. There's the small crocs. You can see that the mangrove systems at the front are quite a significant sort of habitat for them, where they're holding out in there and not so much up into the main river system. And this might be the reason why there's a lot of larger ones up in that main system. You can see a bit of a pattern happening there territorially. And there's a bit of a cluster of points right up the end. This is where all the commercial tour operators hang out. And this is where all the Barrow fishermen do their netting. So I'd say their animals are being fed, and they understand that there's a constant food source there, and there's been able to hold a lot larger crocodiles in a smaller sort of area. So that was interesting, just looking at that data, quick little snapshot from a management point of view. Andy also put together some more pretty little graphs, but you can sort of see that in the 1978, there was a lot of smaller animals and not many animals, and then later on in the 2015, there's a lot more, and the size of the animals increased, and so the graph's sort of taken more of a slide to the right. So basically, it's demonstrating a healthy population. Another graph, the red, I believe, is the 3 points over the whole river system over the 3 years, and then the blue is the breakdown of each section of the river over that period as well, and that power graph, I suppose you could call it, is another good indication that the species is recovering well and healthy. It's another graph, we'll just skip that one, running out of time, but that's just compared to our rivers, the Prince Regent to the Adelaide River. You can see there's a bit of a pattern, same sort of system happening there as we go. Just quickly there, you've got 3 per kilometre in the Prince Regent and 4.2 per kilometre. Some of the interesting facts is our rivers are holding a lot more smaller crocs, and the Adelaide River is holding a lot larger. And this may be, again, just as the river system is slowly developing or increasing and settling over time and starting to plateau. So it'll be interesting to see what happens over time, whether this increases and we get similar weight and numbers as the Adelaide River does. That's the basic results from the genetic sampling. The Prince Regent species were technically a genetically different species, and from a management point of view, will probably need to be managed in the same, or not managed in the same way as the NT crocs. We need a bit more work to happen on this. So into the future, what does this mean? There's a larger population of crocodiles starting to establish themselves and moving into more of a public area. Just what I said there, that there's atypical environment in the West Kimberley, and so therefore more researches need to be done just to see what impact this is going to have as the human population also increases. So there's a few modelling methods we can use. It's what the Darwin guys use, which is the ratio of favourable wetlands, also the rainfall and temperatures. Skip that one, you can look at that one later. This is a typical nesting habitat there in the East Kimberley. This is what we see here. We don't have these real favourable sort of nesting type areas. However, these seem to be quite successful when you look at our data. Many hatchlings recorded up in the catchments. It's a typical nesting area for Prince Regent. So another modelling technique we can use is the data that's collected from the NT parks. Late wet season, you can see there are a lot of crocodiles caught and in the build-up, and they seem to be around that 2.2 metres and mostly male. This demonstrates to us that it's a lot of young males moving around trying to find territory and breeding, or the bigger ones also pushing them out. So we can use this for management as well now that we start to get some figures. Another system we can use is the satellite tracking, and this would be very handy to do over in the West Kimberley, just to see if there's same results as what they're getting over in Cape York. But basically the animals at certain times of year around the breeding time will cover a large distance and travel and cross over territorial areas. It's just some of it's a website there if you want to have a quick look at some of the stuff that's happening over in Queensland. And I know Andy was very interested in doing some of this stuff here in WA, which we may do later on down the track. So just to quickly like to thank state government and the WAMSI partners and some of the DBCA people, the Darren Moncrief and Shane French, Kay Winston and the crew from PB Warndam. Their support was amazing. Also, I won't thank myself, but, and then also the traditional owners.
Yeah, and on behalf of the traditional owners, me and the Derby crew back up in Derby, I say thank you too for giving us an opportunity to go back to the country, and I think it's opened up a few opportunities for my people and my rangers to go back and maybe one day manage that whole country and look out for what it is. Thank you.